talk to you guys about some of the cool stuff. Earlier today we had our grand opening, so this is a real operating facility, really dealing with individual health, and basically it's ability to give people the information and to empower them with knowledge to actually make changes. And what I want to talk about tonight is some of the science. So like what Mitch and I have to do is we have to do things in a manner, in a clinical trial format that's acceptable to our colleagues. Okay, we live in a world of peer-reviewed literature publication, so there's things that we need to do in a certain format. And it's very difficult to translate that to everyday life, because what we really want to be able to do is take these findings and impact the lives of everyday people, like ourselves, which we are one of those everyday people. So what I want to talk a little bit today about is the Plasmalogen hypothesis for the prevention of neurodegeneration and the treatment of neurological function. And as Erica mentioned, I'm an informed boy, so I like to follow two sets of rules. One is, people may have heard the phrase Occam's razor, and it basically says plurality must never be positive without necessity. That basically says if you have a problem and you have more than one solution to that problem, we're obligated to take the simplest solution. So if you have a crop failure here in Saskatchewan, you can ask the question, what was the reason for it? Was it a micronutrient balance, or just like it is now, it just didn't rain? And so the Occam's razor would say, if you have no rain, the most likely reason you have a bad crop is rain, not micronutrients in your soil. The second question is, Einstein says, everything should be made as simple as possible, but not simpler. Okay, so there's a common sense situation that you don't want to simplify things to the point where they have no meaning anymore. Okay, I didn't have a good crop because the moon was in the wrong phase in May when I seeded. Like, so those are the things that we want to be careful. We stay between these two lines of thought. The whole concept of prodrome sciences and our programs is understanding a prodrome, in that we don't walk down the street and get hit by lightning and the next day have cognitive impairment. There are things that happen in our body before these things occur clinically, and that's called a prodrome. So we have a healthy period of our lives that we're pretty well impervious, and we take advantage of that because we pretty well behave like impervious people. And then certain risk factors occur in our lives, but those risk factors will be there for quite some time before they may translate into a disease. And ultimately, when you get a disease, it is a transition. Let's see if I can work this thing. Yeah, so the prodrome, and then the prodrome at some point translates into a disease. So for instance, if you're type 2 diabetic, you have a prodrome of high blood sugar. And at some point in time, that may translate into renal failure. So then you have the prodrome, which is diabetes, and then you have a disease, which is renal failure. So at this stage, you basically have both high blood sugar and renal failure. And if the disease doesn't get treated, eventually we die. And of course, we all are going to die eventually, of something or another. What we're interested in is curing disease. Okay? And curing disease is understanding this risk phase and correcting this risk phase before it turns into the disease. And I often use simple analogies for individuals. Risk is basically a bald tire, and disease is basically a flat tire. And so if you have a bald tire, you are at risk of getting a flat tire. Now, if you get a flat, now you have two problems. You have a flat, bald tire. And so now you can go and fix the tire. You can put a patch on it or put a plug in it, but that doesn't actually fix the tire. It fixes the disease of the tire, but you still have a bald tire, and you're more likely to get another flat, even though you have a fixed bald tire. And so people think of cancer. They get a cancer and they get a tumor, and they go and they get, the doctor comes in and he cuts the tumor out, and all of that, you're not miraculously cured of cancer. They've just reset your biochemistry, so if you live long enough, you may get cancer again. So prevention of disease is quite different than the treatment of disease. When you have disease, you must treat that disease because it is a problem. But treating disease and preventing disease are philosophically two very different things. So cognition, and we think about Alzheimer's disease, but Alzheimer's disease is really, we conflate things. It's a functional system. Our brains aren't working properly in a certain way. And so some people look at these 5 A's of dementia, which is basically increased cognition. So like when Mitch was talking about, the different measurements, we consolidate these different things. Amnesia, which is the most common, people have memory loss. Anomia, where, what do you call this thing? You can't remember names. Apraxia, misuse of objects, why doesn't this pen work when you're playing with your, and agnosia, what does this taste like? Something's wrong with these feelings. And so it's a combination of things. And aphasia, which is probably what I'm having right now, which is the inability to express yourself through thoughts. Okay, or basically the entire male population. So we think it's biochemistry. Your brain does things and it signals from one neuron to another. And this is essentially the cholinergic system. So when people take Aricept, it's a drug that blocks an enzyme that keeps the acetylcholine levels high. So basically it allows the electrical system of a particular part of your brain. So if you think of your brain in separate parts, this is the cholinergic system, which is a focus of cognitive impairment. Parkinson's, for instance, that could be a dopaminergic neuron. And it's very, very much like different systems in any kind of complex system, like a car or something. So not everything goes wrong at once, but you can isolate those issues. And then these neurotransmitters that get fused out and released occurs through a really strange process called membrane fusion. And this is a process that we've shown over years that are pretty heavily involved in plasmalogens. So if the membranes that Mitch was talking about, Dr. Kling was talking about, have low levels of plasmalogens, this fusion process is much more difficult to occur. So it's resisting the ability. And so a thought takes more energy than it should take. So we've been working on this for quite some time. So I published back in 2007 that these ethanolamine plasmalogens in the blood are a logical causative factor. Okay, and so because we saw these associations, very similar to what Mitch has now confirmed in these large cohorts with ADNI and at University of Pennsylvania. And this was in 5 different populations. We were dealing with the, San Diego, Dr. Sparks from Sun Research Institute, Alan Lerner from Case Western, Dr. Takeda from Osaka University in Japan. So we're seeing this low levels of plasmalogens all around the world. And it looked like as the severity of the disease, the more severe the cognitive impairment in the individual, the lower level the plasmalogens were. And we do fancy statistics that call it an association. But fundamentally, those get lower. Now these are cross-sectional analysis. We don't know, as Dr. King was saying, what comes first. Is this just, sometimes because time creates an element in it. It's how quickly are you declining can confuse your results. So to study that, we look at longitudinal cohorts. And the Rush University in Chicago is one of the most advanced cohorts. People enroll in this program cognitively normal. And it's been going on for about 20 years now. So Dr. Bennett's been running this program where people enroll healthy. They get monitored every year, kind of similar to what we do here. Right? We get cognitive testing, they get mobility testing, get quality of life testing, and they collect blood samples, they store them, they track them. And then as people progress in their life, they go into the Rush cohort every year. They come in for these very comprehensive analyses, and then they donate their brains to the Rush cohort science. They actually have all this longitudinal time, and then we actually can study their brains when they do die. And some will die with cognitive impairment, and some will die without cognitive impairment. And so it creates a very valuable resource. Of course, it's not practical to do this in 1 million people in the province of Saskatchewan, but we need to do these kind of precise studies so that we can kind of then translate it later. So the group that I'll talk about a little bit is 1,262 people at 2 different times. And so we're looking at what happens over time. And the average distance time from their first visit to their second visit was 3.7 years. And then we have no cognitive impairment, mild cognitive, and Alzheimer's disease. So we have a very robust cognitive, 774 people that were normal with mild and those with dementia. But what's interesting is that in that 3.7 years, the incidence of dementia went from 77 persons to 226. So we have 149 new cases. Okay, 149 people that did not have dementia at their first visit over a 3.7 year period became demented. We can ask the question, well, what was different about those? individuals versus the rest of the population, and more importantly, what's good about the people who didn't turn into it. Because this is an elderly population, right? This average age was 81 years old, okay, and 85 at age the second visit. So we're not dealing with young people, we're dealing with people that are in the highest risk category of becoming demented in the regular walk of life. And when we do statistics, we compare like continuous variables. When you have a blood level like glucose or cholesterol, it's not a yes/no question, right? It's not like, yes, I have cholesterol, no, I have cholesterol. The question is, how much cholesterol do I have? And so the value, and is a lot. So as you get farther and farther along, so we use a value called a standard deviation to kind of put the levels in chunks that we can measure. And so this is what plasmalogens look like in that 1,262 subjects. So the median level, most people had plasmalogen levels here. And in this 85-year-old group was about 13% or 14% cognitive impairment already existing. But what this shows is that with each standard deviation increase in plasmalogens, the percent of people that had dementia went down dramatically. Okay, so there was a 50% reduction in dementia with each standard deviation increase, such that people with the high levels were down to 5% dementia, and people with low levels were up to 35 to 40%. So as we get to the different spectra, because sometimes things happen where, like a lot of your vitamins you take, like low levels are bad, but it's not necessarily high levels are good. Sometimes you just need enough. And anything above, as long as you have enough to satisfy a certain need, anything above that is just extra, right? You don't really need it. So some deficiencies that, if you think about scurvy and the limeys of the 1800s so they took vitamin C to prevent themselves from scurvy. That prevents the disease, but high levels of vitamin C don't necessarily get any better. You just need enough that you don't get scurvy. And so then we can ask the question with age, because we know age is a big problem. As we get older, okay, so this is how we graph people with low plasmalogens, people with high plasmalogens, the normal plasmalogen levels, and low plasmalogens. And we can ask the question, what's the incidence of dementia as we get older? And so what this basically says, in an 85-year-old person, okay, with high plasmalogens, it was roughly 5% risk of dementia. Okay, but people with low plasmalogens, it was almost 30%. So it's a really big difference. And these error bars are called the 95th percentiles. Okay, so that means when they're far apart from each other, this is a very robust observation. But not everybody experiences decreased plasmalogens as they get older. Like the average level, like Mitch showed you, goes down. But that's not everybody. That means statistics are kind of funny things. Like we use these numbers to compare big populations for one another. But most of the time, statistics have zero meaning to you and me, because it's the groups, and through these groups we can kind of figure out where the smoke goes and where the fire is. That's what we have as scientists are obligated to do. But sometimes the numbers don't have good everyday meaning to us. So what we can ask the question is, what happens to these people that have high levels of plasmalogens? Because this level is kind of like the levels of 30, 40-year-olds, but they're 85, right? So these are 85-year-olds with young person levels of plasmalogens. And what we do is we do this time to dementia analysis, which we basically say, okay, let's start everybody that has no dementia at time 0, and let's track them for 6 years. And let's see what happens in people that have high plasmalogens versus people that have normal plasmalogens. And so everyone starts with no dementia. When we did that, what we found is over a 6-year period, and this is what we call it's like a survival graph, but everyone started with no dementia. So the people that had no cognitive impairment were all 100%, because everybody started cognitively normal, 85-year-olds, roughly. Okay, but what we found here is that people with high levels of plasmalogens over a 6-year period basically didn't get dementia. Okay, there was very, very low incidence of dementia. And when we do the statistical analysis of that, we found that randomly, if these people were the same as everybody else in the study, they should be the same numbers. And there should have been 30 cases of dementia in this high plasmalogen cohort, and there was only 6. And then the p-value here shows how confident are we that the number is real? And that basically says 2 chances in a million that that might be a false observation. So we're very, very confident that that's a real observation. And so we're dealing with an 80% lower risk, incidence of dementia in individuals that had high plasmalogens at baseline. So these are pretty big numbers. And we talk about mortality. I like to show this graph because it shows you if you can make it past your first year of life, you've got pretty good genes. If you have bad genes, you pretty well die early. And so the rest of us for a good 50 to 60 years, you pretty much have to get hit by a bus to die. Okay, you can pretty well do any stupid thing you want, and you're not going to die. And all of a sudden, all hell breaks loose, and basically so what the hell's going on here? It's like something's going on, but it's not all the same person. Like, we don't all die of the same thing at the same time. It's not like we all go through puberty within a couple of years of each other. Like, this is a death, and this is a much different, broader, diverse, inaccurate phase. And as Dr. Kling was saying, we've measured these molecules in the Saskatchewan population. So this is 990 individuals from Regina, so not too far. I know they're not quite like us, but they're close. And we published this not too long ago, but you can see the plasmalogen levels going dramatically down after your 50s. It is interesting that it goes up, so you can tell your kids that your brains don't actually fully develop until you're 50 or so. It's through myelination. That's why they're pretty well stupid until then. So, but anyways, the point is that we do know we have this very severe drop-off as we get older. But the regular phosphatide ethanolamines that he showed that had the regular linkage to the SN1 position, those basically stay constant across all life. It's these plasmalogens that kind of change as we go forward. And this graph here is, quite frankly, the scariest graph I've ever made. This is, so back in this cohort, since we track these people, and he's been traveling, fast here. It's been a 20-year study, so people are dying. It's an elderly population, okay? And so we have over 700 and some of these individuals over time have since died from their last clinical visit. So when they can come in, they can get their testing done. At some point in time, they get old and they can't actually come in for proper testing. They're no longer capable of completing the Rush trial observation. And that's what's considered the last clinical visit. And from their last clinical visit, the average time to death from that last clinical visit was about 1.6 years. So the visit itself kind of gave us an indication that, it's, obviously none of us live forever, so at some point in time that's going to happen. But we can ask the question, almost very similar to what this question was, but instead of asking for what happened with dementia, we can ask the question, who's dying? Okay, and we have 2 visits, so visit 1, and then 3.7 years to visit 2, and then 1.6 years to death, so that's 5.3 years. And so we can ask the question, what's the probability of dying in the next 5.3 years from your age at visit 1? And this basically says, if you take a look at this graph, this is high plasmalogen population, this is low plasmalogen population. And this bar here is at the same line as this bar here. This basically says that a 65-year-old with low plasmalogens has the same probability of living to their 70th birthday as a 95-year-old person with high plasmalogens will live to their 100th birthday. So it's a really terrible, terrible graph. And so, you're dealing with, an 85-year-old, 20% chance of dying over the next 5 years versus 70% chance. So I can pretty well tell you which graph I want to be on. So the question though is, these low plasmalogens are bad, right? So that's what these graphs are. But is it causation or is it coincidence? And scientists sometimes get trapped in this thing. We get these biomarkers and we think that they're the actual causal agent. And that's when the problem with the amyloid hypothesis is that on this amyloid in the brain, we thought this was some toxic molecule, like a Pac-Man eating your neurons. So we think if we remove this toxic molecule, that miraculously the brains are going to get better. And that just didn't happen. And so causation and coincidence is kind of like saying, you get black smoke coming out of the back of your car, and you're saying, wow, what's that causing? So you can say, well, your piston rings are bad. So every time a piston ring fails in a car, you get black smoke out the back end. And so black smoke is a biomarker of bad engine function. But of course, no one would say, well, all I need to do is put a filter on my tailpipe and hide this black smoke from the world, and miraculously my engine is going to start working better. That wouldn't make any sense, right? But obviously, if I found out that someone put diesel in my gas car and I got a bunch of black smoke, well, then there's causation for you. So the question now is asking the question, are these plasmalogens just bystanders to some other accident, or are they actually part of the accident itself? And so to do that, we have to look at neurodegeneration. And can we find a model that causes neurodegeneration, and can we stop it from happening? And if plasmalogen can stop it, then we know that they're not just sitting there at the side of the road watching the accident, but they're actually part of the problem themselves. And one of the things we did is look at Parkinson's. So we also studied Parkinson's in this study. These are all these numbers, same people. And we found that low levels of plasmalogens were also very strongly associated with Parkinson's. So odds ratios less than 1 is good. I won't get into this in great detail, but bottom line is that people with high levels of plasmalogens had a lower likelihood of Parkinson's as well. So we're not dealing with just one particular neurodegenerative disease. We're dealing with neurodegeneration in general. And so they're strongly associated with an increased risk of Parkinson's disease. Now, Parkinson's is unlike Alzheimer's disease. It affects these dopaminergic neurons, and they project into the striatum. And so you guys probably all heard of awakenings and the L-DOPA hypothesis. L-DOPA is one of the most miraculous drugs we've ever discovered. So people that have early stage Parkinson's, L-DOPA, which is basically a bioprecursor of dopamine, pretty well cures Parkinson's during the early stages. It doesn't prevent those neurons from gradually degrading as time goes on, but in early stages, you can take Parkinson's in individuals that are completely normal at that point in time. Now, we use Parkinson's because we can study it. And it turns out a group of kids were trying to make synthetic heroin back in the 80s. And the United States regulates drugs of abuse by drugs that are on a certain list. And so if you're a fancy chemist, you can manufacture an analog of a drug and take it, and you're technically not breaking the law. So these kids were trying to make a synthetic version of heroin, trying to make this MPPP. Unfortunately, they weren't very good at chemistry, and they created a drug that had a toxic byproduct called MPTP. And they all gave themselves Parkinson's. So it's a sad case for them. Okay, but now in the 80s, since the 80s, we've now had this neurotoxin that selectively hits these dopaminergic neurons, and it degenerates dopaminergic neurons that cause Parkinson's. So we can actually create Parkinson's in an animal and ask the question, can we modify that? And how we did that is we take mice and we give them plasmalogen precursors to augment their natural levels to higher than natural levels. And we give that plasmalogen precursor 5 days just to get accumulated in the body. We provide this dopamine toxin. We let the toxin work for a few days, and then we find out whether or not the plasmalogen treatment prevented that neurotoxin from killing the dopaminergic neurons. And it turns out it did. So the striatal dopamine transporter, this is a biomarker of those dopaminergic neurons. And this is the vesicular transporter. And basically you can see here that the untreated animal had normal levels, and then MPTP caused neuron degeneration. And 10 milligrams per kilogram, and then 50 milligrams per kilogram. So at 50 milligrams per kilogram, we ended up getting complete neuroprotection. So we basically completely prevent neurodegeneration from a toxin. So we're giving this animal a toxin that's supposed to kill these neurons, but if these animals had plasmalogens in their blood beforehand, the neurotoxin didn't have any effect on them. And so that's how we look at prevention of a disease. The next question is, what happens if you actually have a neurological disease like Parkinson's? Can it actually improve neurological function in any way, shape, or form? And to do that, we look at monkeys that have, we published all this work. These are Parkinsonian monkeys, so they're not prevented, so the dopaminergic neurons are already gone. So 85% of the neurons are dead, so they have about 15% of the neurons left. And they do respond to L-DOPA, But people with Parkinson's, L-DOPA works very good for a long period of time, but eventually it stops working because it doesn't prevent the neurodegeneration. And eventually there's not enough remaining dopaminergic neurons for the L-DOPA to actually help. And they also start getting weird movements because L-DOPA eventually has some side effects, so they're called dyskinesias. So anyways, what this basically says is that we treat Parkinsonian monkeys that have dyskinesias, and within 2 days of the plasmalogen precursor, these dyskinesias were down in a statistically significant manner. And it stayed down for quite some time. So here, the plasmalogens are actually doing something. They're biologically active, they're neurologically active. And finally, the third disease that we focus on is multiple sclerosis. And multiple sclerosis is a slightly different disease from Alzheimer's and Parkinson's. Alzheimer's and Parkinson's are neurological diseases. The neurons die. So the cholinergic neurons in Alzheimer's disease, the dopaminergic neurons. Multiple sclerosis is a disease of wiring. So this is what your brain is wired like. So this is the neuron. So this is the actual, dendrites come in and the cell body, and then it sends a signal down the axon. So the axon is like the wiring of your car. Okay, it sends a signal from your ignition or your lights or whatever it is to the place where it's supposed to go. And that's how your neurons work. But they're protected, just like the plastic coating on your wiring of your car protects your copper wire on the inside. These oligodendrocytes create this myelin sheath, which is called white matter, that protects the neurons, axons, and allows that signal to propagate. So just like if you had overheated engine and all of a sudden your wiring harness got all melted to crap, and then you have your wiring just starts, breaking down, and you get basically not working. I'm thinking of the right word. Short-circuiting is the word I was thinking of. That's what happens in multiple sclerosis. So what happens in multiple sclerosis is there's an inflammatory event. So unlike heat or fire in a car, people with multiple sclerosis get an autoimmune disease, and these inflammation basically eats away at this protective coating. And if it eats away far enough, it then kills the neuron underneath it. And to study that, we use a drug called Prodrome, and basically this creates a very specific neurotoxicity for oligodendrocytes. And I'll jump ahead here. So if we give animals for 6 weeks, we can create quite a significant amount of demyelination, which is the pathophysiological observation in multiple sclerosis. And so if we treat the animals for 7 weeks, but the last 2 weeks, weeks 6 and 7, so we give them the plasmalogen precursor, all these are biomarkers of remyelination. So multiple sclerosis is a disease, early stages, it's relapse remitting. So people initially get a symptom of muscle fatigue, loss of balance, and things, and then they go in and get diagnosed, and the MRI shows lesions typically. And then it goes away. It goes in a remitting phase, and they don't know why it goes away, and then they have to kind of wait for it maybe to come back again. So it's called relapsing remitting. And as time goes on, eventually the relapsing remitting turns into secondary progressive, and the remitting phase never comes back again. And so that's called remyelination. So basically this shows at the last 2 weeks of demyelination that we can improve remyelination. And the last part is what happens if we give the plasmalogen precursor at the same time as the white matter toxin. And this is what a regular diet looks like. It's hard to see in neuropathological slides, but it's very good cellular formation. This is what a myelin sheath should look like. And in the Prodrome administered animal, it's all disconnected. There's no integrity here. This is what shows up for a myelinated lesion. And then if we provide the precursors, we basically completely prevent demyelination. So the plasmalogens are actually doing things. And so in terms of causation or coincidence, the data suggests very clearly that these molecules are related to causation. Okay, and so that's why we want to get this going. We recently published a paper, Dr. Senaki and myself, just last month in April. And this is with ApoE genotype, people looking at genotype. And this is, the only reason I bring this up is it's a simple table in this graph because we talk about these big fancy numbers. But This is the percent of individuals with dementia in ApoE3 carriers. This is a big risk factor for Alzheimer's disease. E3 carriers and E2 are somewhat protected, but these are 3 different cohorts. And people with high plasmalogens, normal plasmalogens, and low plasmalogens. And so the percent dementia in the E4 carriers goes from 37% to 20 to 14 with increasing plasmalogen levels. In E3s, it goes from 30 to 12 to 6. In E2s, which is protective, it goes from 22 to 6 to 0. So you actually have people that fundamentally have almost zero risk of dementia. So the point of the story is it's possible. Okay, it's not inevitable that everybody gets this disease. And what we need to understand is, how do we get more people into that category? Okay, that's the goal. It's fun. Not fun. It's we often focus on the negative side. We focus on the disease. Why does someone have a disease? But Alzheimer's is a weird disease because most of us will get it if we live long enough. So what's really the interesting part of Alzheimer's is the people who don't get the disease. They're the odd ones. Okay, the normal person is a person, if they live long enough, they will get the disease. So the odd people are the ones who don't get it. And the question is, why don't they get it? And there's interesting interest with the ApoE2, it's the cholesterol transport, and so on and so forth. So that's my last slide. We're going to look at community trials. So we have precision-based trials with UPenn. But at a community level, we need to be able to do things in a much more larger scale, simpler plasmalogen precursor that's very inexpensive, easily administered to larger populations, simpler test. And so the Prodrome Health Center is designed to develop functional tests that are not threatening. Like the Rush University has 19 cognitive. It's like a whole day to get poked and prodded and examined. And quite frankly, I would stress the hell out of me to go through something like that. And for these people, they're amazing that they've stuck through these programs for like 20 years. But that's not practical for the real world. And so we create functional tests at Prodrome Health that are non-threatening. They're designed not for me as a scientist, they're designed for you as an individual, because you're the one who uses the results. It's not about us using the results. It's saying, okay, how can I get my cognitive assessment done in a manner that doesn't scare me? Okay, so I can get an assessment of it. My mobility, my grip strength, how quickly can I walk 10 meters, my posture. These are simple, basic, everyday things that mean actually have a very important meaning in your quality of life and how your value of your life goes forward, as well as your quality of living, like your mood and other things. So these are things that are designed for you to say, okay, this is where I am today. The blood testing also measures lots of things to say that we can look at what biochemical issues are in your body that you can't feel or see. And then the next step is saying, okay, now that I understand my current status, what interventions have been scientifically proven? There are diets, there are exercise programs, specifically resistance training programs. There are things that actually work, okay, and they're measurable in their ability to work. And the trick is getting that into a model that an everyday person can use. And we run these trials, okay? There's trials every month or two. There's publications coming out, this does this and this does that. But then the researcher goes on to the next project, and then the next project. And none of this stuff, or very few of this stuff actually gets translated into everyday work, because that's the hard work. Okay, this is the hard work of actually working with real people and getting real things done. So the point of the Prodrome Health Center is to take this scary science side and put it in a format that individuals can use. And then they say, okay, I make a joke because I can't remember how I felt last week, let alone 6 months ago. So tracking your performance and understanding if you feel better, sometimes it's hard to know that until you look back with your consultant and says, you know what, this is what you were saying was going on in your life 6 months ago. You actually are seeing improvement. So there's the functional, even though you may not remember where you were before. And conversely, if people try different supplements or try different other programs in their health, it might be doing bad things for you. You might want to know that. And so the ability to actually have that feedback is what we're trying to accomplish here. And fundamentally, we're interested in population statistics in the province. How do we actually reach the 13,000 individuals, okay, that have dementia in the province, and the 3,000 individuals who will get dementia in the next 12 months? Okay, how do we get to those people? And how do we bring those numbers down? Okay? And do it in a way that a population can actually measure it. So that's what we're all about. And the interesting about the Plasmalogen program, even though that's the starting point, it's really the programmatic part of a community, and how do we interact and actually make real change happen. So that's the end of my presentation. Thank you very much for staying around. And we'll have questions for Rich and myself, if anyone has thoughts or ideas.
So what's the next step?
So the next step is we're doing the manufacturing, and we're doing a specific molecule for the biomarker trial, and then we're manufacturing a larger, more user-friendly plasmalogen precursor for clinical trials. And so our goal is to get ready to roll this thing out. So our next step is to get all the wheels turning in this facility. As you can see, it's a pretty big facility. We can handle a lot of people here as we go forward. And we need to get that infrastructure in place, working with people like Greg to deal with the international marketplace. I don't know if Kevin's back. So Kevin from the Buck Institute and Openome, he works with us in terms of, so yeah, let me do a little bit of the interaction. So Prodrome Health has to do physical hand-holding, okay, to get this stuff done, because in the whole part of the community program. That's why Jana's here, and this used to be the medical detox. And we're talking about modifying behavior in a positive manner and sustaining that positive modification over time. And the only people who have successfully done that long-term in human health is the drug and addiction community. Okay, people who have addictions, okay, these addictions destroy their lives. They destroy their relationships, and they ultimately kill them. So it's but changing behavior in a positive way is not easy. It takes community, it takes knowledge, and it takes dedicated staff to help that process. And so we're taking that type of commitment to understanding positive lifestyle changes that actually have meaningful benefit here. So that's what Jana and the team here at Prodrome Health has the experience of doing. We are also looking at, so Mitch and I focus on the clinical trial and actually the precision of actual mechanisms. We do have our colleagues that we answer to. There's certain precision types of trials that we're obligated to do in certain manners, and that's what Dr. Sanaki and I and we all do. But Kevin, with Openome and Open Cures, can take this to a much more global scale of people self-organizing, the blood testing, we have relationships with people, the APOE4 users group, and so on. But create an ability for people to get blood testing. They do their own self-trials, they share their metabolic data with each other, and they figure out what's going on. And that's what Kevin and Openome and Open Cures is all about, is accelerating the cycle of knowledge, intervention, and back and forth. And then Greg Goodenowe heads a network called the EQ Network. And this is a different type of global infrastructure. So he's based out of New York. We were just out of Berlin in terms of the ability to create a community of like-minded individuals that are interested in Alzheimer's and other neurodegenerative diseases, but may not live here in Saskatchewan. And so they can contribute and be part of this program, okay, knowing that what we learn here will be translated elsewhere. And we need to build that community, and it's also part of the financing, the crowdfunding of this model, because our ability to offer services in some of the trial programs to people who can't afford them. So we're looking at this as a population health, and we need to find mechanisms to do that. And there's really novel ways of doing that with Greg's effort, and also the way to protect everyone's personal information, because this information is highly protected. Individuals have their own access, they have their own ability to track it. So anyways, that's how we fit into the whole aspect of it. So
I have just a question, but I haven't actually asked you this before, but reaching the 13,000 people who are currently suffering, and that's just Saskatchewan, you think that if they knew about this, or if their doctors knew about this, they might actually consider telling one of those 13,000 people that they deal with, because they're all probably seeing a physician.
Correct.
What are any plans for reaching out to the medical community or government?
We will systematically. I think our focus primarily is on the individual.
Yeah.
Okay, and they are the ones that will, it's from the individual to the doctor, not from the doctor to the individual. Okay, and so we're growing from that perspective. Obviously, we're interested in working with all individuals on that. And right now, the obligation, the responsibility is on us to prove these things work. Okay, when I published this in 2007, okay, that's 12 years ago. Okay, 3,000 people a year roughly die. So the entire population of Moose Jaw has died of dementia since from there today. Okay, so it's a time to get this out there. And so we need to figure this out, we need to do it. The nice thing about it, we're dealing with a very natural product. We'll do the real science with on everything, but we have to do this. It's our responsibility to get to a certain level, and then at that point in time, the conventional communities will listen. And then that's why we've chosen Saskatchewan, because we can actually make a difference on our entire population. We can bring this 13,000 down to 12, down to 11, down to 10. And so when we come back here in 3 to 5 years, maybe this number, we have one of the lowest rates of dementia in the Western world by simple community hard work. It's doable, the data's out there. It's just a matter of actually doing it. And that's like the hard part. And that's what we've been thinking about and designing here, is to do that hard part. And then once we can solve the hard part, then the rest of the world will pick up the pieces and go from there.